Are we going to do questions now, later, never? What would you like to do?
Yeah, we are open for questions now. Anyone?
We're going to get your light.
Thank you so much for sharing your paths from students to clinician scientists to Nobel laureates. It's quite inspiring. I have a question in this area. In this era, research is rapidly changing since some technologies such as AI. Would you mind share some tips or suggestion to young researchers who are at the very early stage, how to stay at the forefront in this era? And would you mind to share some tuning points in your career which embracing change made a big difference? Okay, thanks.
Yeah, it's a wonderful question, and I could go in several directions with it. First of all, I loved clinical medicine, but I never felt like I was playing. I never felt guilty someone paid me to do it. Once I started doing science, I was having so much fun. I felt guilty that people were actually paying me to do it. I don't mind that they pay me, but I did feel a bit guilty that they pay me. So I tell you that because part of the secret is find something that for you is so much fun, you can't wait to come to work the next day. And I start with that piece of advice because I'm always a little reluctant to project my interest onto other people. The key is to find your interest. Now, I'm looking at my colleague, Tom Rappaport. I think he would agree. I was trained to be something of a reductionist biologist. I like solving puzzles, and I like trying to understand how things work. That's a somewhat different type of science to what we'll call some types of systems biology, where you use powerful technologies, you get very big data sets, and you go mining. Now, those are complementary, and for some people, that's exactly the right kind of science to do. It's not my kind of science. And I would caution there is this tendency sometimes to look at the next shiny technology and say, I want to do that because that's the next great thing. For some people, again, that's the right answer. But one of the many things I learned from David Livingston, in my view, every good experiment or every good line of investigation starts with the question. If you start with the technology, you quickly run the risk of becoming a hammer just looking for nails because all you think about is my technology. I think it's more fun to start with puzzles, conundrums, unexplained phenomena, and then try to figure out how they work. I've yet to see one of these systems biology heat maps or UMaps or whatever maps you want actually explain to me how it works. So again, find your own thing that makes you happy, but I would warn against chasing the latest, greatest technology. And with respect to AI, I think at least for the foreseeable future, we're going to work with AI. But I think the term I like or I've heard is AI as your co-pilot. I hope we're not ready completely to turn everything over to AI. So maybe using AI as a co-pilot.
Thank you. Any other questions?
Is there a question? Maybe a question over here, or do we have time?
Professor Kaelin, thank you very much for the wonderful talk. I have a question about the hydroxyproline. So I wonder if there is any specific mechanism on how the hydroxyproline or elicits the pathways to leading to the cancers. Thank you.
Okay. Well, a couple of things. So in the interest of time, I didn't show the crystal structure, which really helped us understand why VHL would only bind to HIF once it had undergone that prolyl hydroxylation. So the prolyl hydroxylation is simply bringing VHL and HIF together so VHL can destroy HIF. So the biology itself is downstream of HIF and the genes that are turned on by HIF. But one of the many puzzles in my field is why, for example, does VHL loss cause kidney cancer, but not, for example, colon cancer or lung cancer or breast cancer? It probably does have to do in part with this important role of HIF2, because HIF2 is not even expressed in many other cancers. When we talk about HIF, we often think about HIF1, because HIF1 is ubiquitously expressed. But actually, in most cells, when cells are hypoxic, HIF1 actually causes them to proliferate less. because you're trying to conserve energy in ATP because you're not getting enough oxygen. HIF2 is a little bit odd. HIF2, at least in the kidney, can make the cells proliferate better. So we're trying to understand more and more the differences between HIF1 and HIF2 and how those differences are achieved.
Thank you. Now there is a little gift for you.
thank
Please.
Okay.
Yeah. and
Follow instructions.
We'll take a picture. Now let's move to the second one. So the second speaker is Dr. Novina. Dr. Novina is an associate professor of medicine at the Dana-Farber Cancer Institute and Harvard Medical School, and an associate member of the Broad Institute. His lab has made several discoveries into the fundamental biology of non-coding RNAs and their dysregulation in cancers. His lab also developed platform technologies to discover and drug non-coding RNAs, build an epigenetic editing tools for targeted DNA methylation, and engineer the Boolean logic gated system for cell therapies. Dr. Novina is a serial entrepreneur and co-founder of 6 companies that accelerates the transition of biological and cell-based tools to the clinic. He is a recipient of numerous honors and awards, including the WMCAC Distinguished Young Scholar Award, Department of Defense Idea Award, the NCI Director's Provocative Question Award, the National Science Foundation Collaborative Research Project Award, and the prestigious NIH Director's Pioneer Award. Please take it away, Dr. Novina.
So I'd like to begin by thanking the organizers for the generous opportunity to speak here today. And also to say, reiterate what both the first 2 speakers said, that we are stronger by having our foreign scientists contribute to this community. And I hope that all of you stay strong in this time. All right, so I'm going to begin by telling you a little bit about long non-coding RNAs. It's a little bit of a digression, so I'll do a 1-slide overview of key features about some of the biology of non-coding RNAs, long non-coding RNAs. So what's interesting about long non-coding RNAs is that the majority of the human genome is transcribed, and much of it by RNA polymerase or RNA polymerase II. So many of these transcripts, they have all the molecular features of a protein coding gene. Most of these mRNAs, they're capped, spliced, and polyadenylated, and the majority of long non-coding RNAs too. In fact, many long non-coding RNAs have AUG sites, ribosomes assemble, and it's still a mystery why many of these RNAs aren't translated. So many of these RNAs, by definition, they're 200 nucleotides long. They turn out to be poorly conserved, and they tend to be low abundance. And for many reasons like that, because they're poorly conserved and low abundance, many people will start to say they can't possibly be important. And I'm here to tell you that actually they are very important, and inhibiting or changing the expression of these things actually has a lot of biological outcomes. So many of these long non-coding RNAs, they arise late in evolution. Most of the long non-coding RNAs expressed in humans are not in mice, which is not in worms, yeasts, and flies. And so it becomes very hard to say, well, if the long non-coding RNA is in an earlier model organism, we know what it's doing in a higher organism because they don't exist. And because they're poorly conserved, it's hard to sequence align them to have sort of a sense of where they belong in the hierarchy and function. And RNA protein folding, of course, still, with even all the advances that are there, is largely based on lowest enthalpy, lowest delta G. And most of the functional RNAs are not the most stable RNAs. In fact, RNA's real function comes from the fact that they assume many different intermediates and bind to different biological molecules, most especially proteins. And so not the least energetically stable is not necessarily the one that is most stable, or most active. So this makes it very difficult to study long non-coding RNAs, but it also makes them very interesting to study, and they also make them tractable drug targets. So many long non-coding RNAs have been implicated and associated with a large number of diseases. They've been most tightly associated and clinically associated with disease outcomes in cancer, but they're also associated with neurodegeneration, autoimmune disease. There's a growing literature for their involvement in fat biology and metabolism. But I would also like to caution, as Bill said in the last slide, causation is not directly a consequence of things that are co-associated. So there's a large number of papers that associate long non-coding RNAs with the disease, but it's very hard to figure out what roles those long non-coding RNAs play. So how do you understand what long non-coding RNAs do and how they're related to physiology and how their dysregulation relates to disease? So it's very difficult to do. It's very difficult to understand the biology of long non-coding RNAs when you have low abundance, poorly conserved entities that are expressed uniquely in certain tissues of certain organisms and not in others, not in other tissues and not in other organisms. So one of the insights of my laboratory, sort of a central tenet of how my lab has operated over the last 20 years, is to understand the biology of long non-coding RNAs or all non-coding RNAs by discovering the proteins that interact with them. So if you understand, for the most part, we know what proteins do functionally. We know that a protein might be involved in epigenetic regulation. So you have polycomb-associated complexes that are involved in chromatin accessibility. So these long non-coding RNAs are involved in DNA accessibility, both in cis and trans. There are proteins that are involved in transcription, splicing, are in protein export, and in translation and signaling. So if the protein is involved in epigenetics, for example, and the RNA binds to it, well, then most likely the RNA is involved in epigenetic control. And similarly, if a protein is involved in, let's say, proliferation, sorry, I'm gonna do this with my finger here, with proliferation, metastasis, or EMT, then the RNA is involved in that process, okay? But as I just said a moment ago, causality is not a direct consequence of their co-association. So you want to actually add more function or more insight into annotating real causation to two things that are co-occurring, all right? And so what my lab has done is we try to find two things that are associated, just two things. One protein is associated to a disease, and RNA is associated to the same disease, and they interact. It becomes less likely that those two things were happening by happenstance. If you disrupt the interaction and you have a reversal of the biology, then you now begin to annotate these interactions to causation. Okay, and that's one of the, so I started, as the introductory remark said, I've started 6 companies. One of my companies called Next RNA is doing just that, finding small molecules that disrupt RNA protein interactions that cause disease. All right, so these are the challenges up here that are associated with studying long non-coding RNAs, and I've already alluded to most of them. They're low abundance, poorly conserved. RNA folding algorithms don't tell you too much about their biology. And if you try to do biochemical pull downs, trying to figure out which proteins interact with those RNAs, most of the processes are inefficient. They're certainly biased because you're choosing the RNAs or proteins to pull down. They're low throughput. And most frequently, you're enriching things that are either the most stable or the most abundant, or both. And that doesn't mean that the weaker or transient interactions aren't important. In fact, oftentimes they are the most important. So my lab has worked on and developed several different technologies to systematically discover which proteins bind to an RNA of interest. And so 3 of them are shown up here. One of them was developed with the Vidal lab at Dana-Farber. So it's a modification of yeast 2 hybrid. So you can split a transcription factor into a DNA binding and an activation domain. When you have 2 proteins come together, you get a transcriptional activation. You can modify this to so that one of the domains, the DNA binding domain, has a high affinity RNA binding protein with a known RNA sequence. You can attach to that an RNA of interest, and then you can screen the orpheome and discover which proteins bind to an RNA of interest. And this is a very efficient, high throughput, low cost way of discovering the commonalities amongst the proteins that bind to an RNA of interest. A second technique that was developed in my laboratory is called RADA. It's basically a modification of the Signosis transcription factor array. In this case, we take an RNA of interest, we put an epitope tag, you could pull down on that RNA, and then you elute the proteins from the precipitated RNA, and you add biotinylated DNA probes. These correspond to transcription factor binding sites. And then you could basically do a wash, put it on a transcription factor array, and wherever you see one of the wells light up, that's going to correspond to a DNA sequence. And then you can then basically by abstraction say that there must have been a protein that binds that factor associated with my RNA of interest. So that's a second way. A third way is a computational way where, typically when you look for associations between transcription factors, for example, and their target sites, you have transcription factors on the x-axis, you have target sites on the y-axis, and you're looking for, co-associations, either positively or negatively. You can add a third factor to that RNAs and find which factors are associated with which targets are associated with a transcription factor dependent on a long non-coding RNA. So we've done that for one of the long non-coding RNAs discovered in my lab called Slinker. And we've discovered, for example, for a large number of genes in melanoma, there are many different target genes that are co-regulated or seem to be co-regulated when E2F1 is expressed and Slinker is overexpressed. These genes seem to be co-regulated. Okay, and you could look across many patient samples and see there are common genes that are affected by both, and those happen to be enriched in genes that are important for melanoma progression. So this gives us a sense that one of these factors, E2F1, is involved in mediating the effect of this long non-coding RNA Slinker. So there's computational ways of discovering this. These are the experimental ways. So yeast 3 hybrid has identified that E2F1 binds to Slinker. This is RADA, where we've done the transcription factor array with pull down, and you have certain regions that are more conserved. Conservation in long non-coding RNA space is a little bit difficult to describe. I'm not going to go into the details right now, but there's a certain region that seems to be related to more than other regions. If you delete that, you lose this interaction. So you have a lot of different independent metrics for saying that in a hypothesis-driven way and then in a global computational way, E2F1 is mediating the effects of this long non-coding RNA called Slinker. So my vignette today is going to focus just on this one particular interaction, but this is generally applicable to any other long non-coding RNAs when you start to analyze them. And I can give you many examples that are also in my laboratory at another time or during the break. So here's a one-slide overview of key features of melanoma that you need to keep in mind for today's purposes. So melanoma turns out to be a relatively rare skin disease. It constitutes 1% of skin cancers, but it's responsible for more than 80% of skin cancer deaths, okay? And it's because melanoma is one of the most genetically unstable cancers. There's, many of the talks at Dana-Farber, you hear discussion of cancer as a disease of the genome, and you have all these alterations, and you have multiple different ways for cancer to evolve. Well, it turns out that melanoma has been a very difficult cancer to find targets for. You could find molecular targets and knock things down, only to see the cancer come back 3 months or 6 months later. But it also turns out that's one of the reasons why melanoma tends to be more sensitive to things like checkpoint inhibitors, where you engage the immune system, because it tends to be more immunogenic. But all this is to say that melanoma is one of those cancers that is, it's very aggressive if left untreated. And if melanomas escape local, when a melanoma converts from in situ to metastatic, it becomes very, very difficult to treat. So it's good to understand what mediates that transition from a melanoma in situ to a metastatic melanoma. So here's one study where I'm going to describe for you sort of a little twist on how we think about oncogenes. Oftentimes you think about oncogenes or tumor suppressors. Oncogenes go up, tumor suppressors go down, if there's mutations. Many people have described both protein coding genes and long non-coding RNAs as oncogenic or tumor suppressor. Here's a situation where we looked at melanomas in an unbiased way across the Cancer Genome Atlas. So this is just all different cancers, all different melanomas, different stages, different sites, different sexes, different ages. And we just asked a simple question. If we take the median split of 2 known melanoma oncogenes, this long non-coding RNA and E2F1, they're 2 established melanoma oncogenes, what's the effect on survival? And so what you see, this is a Kaplan-Meier curve, just splitting these into 4 quadrants: highest E2F1 and highest Slinker versus low Slinker, high E2F1, high Slinker, low E2F1, or low and low in both. If you have low in both or high in either one alone, the patients basically trace with the norm across all these cancer data sets. Only when you have both high Slinker and high E2F1 across the normal survival do you see a worse overall progression. So this is another metric that would say it's neither the protein by itself or the RNA by itself, but its interaction that is helping drive disease. Okay, so these are multiple different ways of looking at whether a complex itself is oncogenic. So my lab actually, we wanted to interrogate the biology of this interaction a little bit more deeply. And so here's a modification of that yeast-3 hybrid system I was telling you about a moment ago. Instead of screening an entire long non-coding RNA, we decided to chop it into pieces and screen for which section of the RNA is responsible for the E2F1 interaction. And so this is actually, it's a 2.5 kb long non-coding RNA. We chopped it into about 250 nucleotide segments, and then we screened for interactions with the specific protein E2F1. And notably, we found only 2 fragments from this RNA interacted with E2F1, fragments 2 and 3. And when we did a deep dive and looked more carefully at those segments, what we found is that within those segments, there was the RNA analog of the cognate E2F1 DNA binding element. So it's interesting, most transcription factors aren't thought to bind to RNAs. Now we know that that is wrong. Recent data have shown that actually transcription factors are enriched for RNA binding. Rick Young has found that transcription factors are especially enriched through an arginine-rich motif, but not through its cognate DNA binding element. So the DNA binding element is thought to be separate from the RNA binding ability of most transcription factors. What was unusual about this is that there were 5 sites within these 2 segments, and each of these had very close sequence similarity to the known DNA binding element. And 2 of them, EF2 and 3, I'll show you in a second, were especially close to each other. So we wanted to test independently whether these segments actually bound to E2F1. So this is an RNA electrophoretic mobility assay. So it's simple, you label an RNA with an element, you add purified E2F1. So this is really reductive science. Does the transcription factor actually directly bind? And this is an unambiguous test. So the answer to this is yes. And each of these bindings are competing or competitive with an unlabeled RNA. Because they're close in space and RNA binding to proteins is thought to be cooperative, we tested whether it was cooperativity. And here's a slide that basically says, yes, if you have each site that's binding by itself, by adding them together, you get better. So that happens for binding sites 1 and 2, which are close in space, for 2 and 3, which are close in space. And it actually turns out, for reasons I'm not exactly sure why, it didn't happen for sites 4 and 5, which were also close in space, but they didn't show this property of cooperativity. But interestingly, this was very robust binding between sites 2 and 3. They are 60 nucleotides away from each other, and they're in antisense orientations to one another. So this segment actually folds into a nice hairpin. And so this is something that looks much like what a DNA binding element might look like. So we wanted to test whether this RNA element has a role in mediating the biology of E2F1. So E2F1, it's involved in numerous biological processes. It's most notably, associated with DNA replication, but it's been implicated in that, DNA damage repair, self-renewal, autophagy, migration, development, and differentiation. I'm going to focus your attention on both proliferation and invasion for the purposes of this talk. And so in this experiment here, we demonstrate that both invasion, melanoma invasion, and proliferation is driven by E2F1. By simply expressing this long non-coding RNA in a melanoma cell, you can drive its invasion and its proliferation. And we realize that the effect is mediated by E2F1. By knocking down E2F1, you block E2F1 effect on both invasion and proliferation. But it's notable, the increased effect on proliferation is further reduced by ectopic knockdown or knockdown of E2F1. So this is, again, strongly associating the effect of Slinker to a specific protein mediating these diseases. Okay, and so the next experiment we tried was to take a modified version of this binding site 2+3 and add it into melanomas and see if we can block ectopically the effect of overexpressed Slinker. And of course, the answer is yes here. So this is increased invasion. If you have a blocking oligo that mimics the binding site for E2F1, you dominantly sequester away E2F1 from the Slinker, and you block the invasion and the proliferation. So again, this is a way of functionally annotating the complex, not the individual proteins or the RNAs to oncogenesis. This is a similar experiment now in vivo. So this is a mouse model of melanoma metastasis. So in this mouse model, you luciferize a melanoma, you stick it in the tail vein, and the most common site for melanoma metastasis once it's in the blood is in the lungs. And in this case, we used this E2F1 binding blocker, And what you see is that you have complete abrogation. So the melanomas are injected in the tail. They typically go to the lungs. And here, using this blocking oligo prevents migration of these melanomas into the lungs. But notably, this oligo does not affect the levels of E2F1 protein or the levels of the endogenous RNA. So again, this is consistent with the RNA protein complex being oncogenic, not the individual entities which are both upregulated, but not having the effect when they're not in complex. All right. So I'm just going to go a few more slides and give you a deep dive into some of the biology and getting into some of the atomic level interactions between this RNA and this protein. Because like Bill, I'm also by nature a reductive scientist more than I am a global scientist. In this case, again, we're going to return to the RNA EMSAs, the electrophoretic mobility assays that I learned back in the Pleistocene age in the 1980s in Bob Rader's lab. And so here I'm showing you that if you label DNA to E2, so this is just pure DNA, purified E2F1. If you can compete the E2F1 binding with a DNA oligo, a cold oligo. As you add RNA, you can begin to compete E2F1 binding to DNA with roughly equal molar RNA levels. But what was surprising is if you reverse this, if you label the RNA and first bind E2F1 to the RNA, and then try to compete it with cold DNA, it turns out that the RNA binding is far more stable than the DNA binding to E2F1. In fact, this is a 1,000-fold molar excess over here of cold competitive DNA. So this was actually quite surprising for a transcription factor binding so well to an RNA of interest. So this suggested to us, because these were competitive directly, that the E2F1 was binding to the RNA through the cognate DNA binding domain because, got it, 5 minutes, was directly competitive. So what we wanted to do was take an atomic look at this. And to do this, we wanted to look first at the crystal structures. And I went to talk with my, my closest colleagues who have expertise at doing structural biology. And I begged for them to help me make a crystal out of E2F1. And they looked at this molecule, which was very large and very poorly behaved, and said, I'm not going to touch it with a 10-foot pole. And they said, we're not going to do crystal structures because E2F1 is a poorly behaved protein with large areas of unstructured regions. And people have actually tried to make crystal structures and failed for E2F1. But instead, there is a crystal structure of E2F4, which is a little bit better behaved protein. And specifically, somebody's looked at the DNA binding of E2F4 with DNA. So in molecular dynamic stimulations, what you like to do is start with a structure that is very, very close and try to model how close these interactions are. And then you could relax the constraints and see how much the molecule breathes to look at how the protein and the RNA or the protein and the DNA would move. So we did this for E2F4 and looked on top of that, model on top of that, the E2F1 DNA binding domain. So the E2F1 DNA binding domain has 60% identity and 84% similarity to the E2F4 DNA binding domain. And when you could see this is overlapped here on the bottom right, the root mean square deviation is quite close. So this suggests that the model is very, very good. And what I'm showing you down here on the bottom left side is that when we label RNA, that E2F1 does bind to RNA through the DNA binding domain. Okay, so this is actually a sanity check to say what we're predicting is correct, that the RNA is going to bind through the DNA binding domain. And so with this model built and with these good constraints, we just decided to run MD simulations. And I'm showing you on the left an MD simulation of E2F1, which is in brown down here. So this is the key loop that is interacting with the E2F1 cognate DNA binding domain. Cyan is DP1. So it's been well understood for years that E2F1 typically binds to DNA and is stabilized by interaction with DP1. So it's a heterotrimer that interacts, very stable. On the right is E2F1 binding to the RNA, that 60 nucleotide element I was showing you. So this is E2F1. You can see the loop is the same position. Here's E2F1 loop. Over here is E2F1 loop. We can't have DP1 on this model because the RNA moves in such a way that binding to E2F1 to the RNA would obscure the interaction site with DP1. So you actually can't model the DP1 binding here. So it almost suggests that RNA is actually displacing its interaction, the E2F1 with DP1. The other thing you should note is that it looks like the RNA molecule is more neurotic. It's moving all over the place, and the DNA is a lot more stable. These are the same number of slides over the same amount of time. So what this is telling you is that the RNA is actually much more flexible and it's sampling more biological space, right? So that's what's fascinating about this, but it actually is consistent that the models show that the E2F1 should be able to bind to RNA through the DNA binding domain. Let's go forward here. And what I'm showing you on this slide, I'm just going to go over it very quickly. The take-home point from this is DNA on the bottom. You can look at DNA is relatively constant and it's binding to E2F1, but RNA assumes a lot of different structures when it binds to E2F1. So it's almost like there's a lot more permissible interactions for E2F1 interaction with RNA than it is for DNA. So just to bring this back to health and disease, there are mutations shown to play a role in the biology of E2F1. A common one in patients with mesothelioma is this R166H mutation. And there are these CHIP analyses across patients with mesothelioma showing there seems to be reduced promoter occupancy with these mutations. So people have automatically assumed that that affected DNA binding. I'll show you that's not true in a second. There's an engineered mutation in E2F1 that was engineered to block promoter occupancy at E2F1 reporter genes like E2F1 itself and CDC6 promoters. These are two genes that have E2F1 binding sites in their promoter. They're reduced with these mutations. So the logical conclusion is these must be blocking the ability of this E2F1 to bind to promoters. When we looked at E2F1 mutations by MD simulations, you can actually look at the contribution of each residue to thermodynamic stability of the binding. And what you see is that both leucine 132 and arginine 166, they contribute only a minor fraction of stability to the E2F1 binding domain. And many studies have shown that the two key residues for contact are arginine 127 and 125, that the greatest stability comes from those two residues. And these two mutations are away from that. Okay, so MD simulations would predict that these mutations do not affect DNA binding. And that's what I'm showing you on the far right, that they do not affect DNA binding by EMSA. Similarly, these mutations are not expected to have a major effect on the overall stability. I'm going to finish in 1 second. I know when I'm also not quite as good as Bill's listening, but I'm getting there. So the thermodynamic stability when you use MD simulations for these mutations, you see that RNA, the stability is distributed over a larger region than is DNA. So you would imagine there's a lot more permissivity to these mutations. That's what I'm showing you in the far right over there. And so this is just a conclusion slide, but basically the whole talk is a conclusion. The point is that, RNA protein complexes themselves can form oncogenic steps. When melanomas are proliferating, they're not necessarily migrating and vice versa. And so this toggle between E2F1 and Slinker might be mediating the transition between melanomas in situ and melanoma progression. And the most important slide are the people who have done this work. Kushani is the lead author. This paper was just published in Cell Reports. And I want to thank you for your attention. And again, I appreciate the invitation to be here today. Thanks.
Thank you, Dr. Novina. One quick question. Okay.
How do you think, how effective do you think the AlphaFold is at predicting the structure of long non-coding RNA and its interaction with proteins?
So the question is, how good is AlphaFold, which is a protein folding prediction, which is actually
And
right?
long non-coding RNA prediction.
So the problem with the AlphaFold in terms of RNA prediction is we don't know exactly the structure of RNAs, but I understand that there are some tools in terms of understanding the key molecules for RNA binding. I would say, because as we start figuring out which proteins bind to which RNAs and which segments, the more you predict the commonality in the domains in the proteins that bind to a particular RNA, the better you will be able to use AlphaFold to make those predictions. But you need to have more information about the domains that are responsible for binding because it's still quite flexible. So
a short question. Great talk. So we know the linker are low copy and the short turnover. So how can you understand the molecular ratio or stoichiometry between the RNA and the binding protein?
So the question is, long non-coding RNAs are low abundance, certainly sub-stoichiometric to many proteins, not all of them. And so how can I understand the
ratio, the molecular ratio between the RNA and protein, how can it function to the regulator to the proteins?
So, excellent question. So if you look at an expression graph for long non-coding RNAs, the most abundant long non-coding RNAs are roughly on the same order, they're very close in expression to the least abundant proteins, like transcription factors. So there can be effects of the most abundant long non-coding RNAs and least abundant proteins stoichiometrically. I would say that in our experience, this is a highly biased answer. In the few that we've studied, when an RNA binds to a protein, you selectively affect only certain activities. So you're not actually changing the effect of all proteins, but you're concentrating the effect of certain factors at, like, for example, certain promoters. But you don't have
to regulate locally.
It could be a local regulation, but it's also that if you're affecting something like a transcription factor, you don't have to have, or an enzyme, you don't have to have stoichiometric levels. Enzymes are catalytic, so a small number of molecules can have an outsized effect on the production of the product. And also a small number of transcription factors concentrated at certain genes would have an outsized effect on those subset of genes. Does that answer your question?
I see. Thank you.
Okay.
Thank you. Actually, we have time for one more question. Does anyone have a question?
You see, you stopped me a few minutes early there. I had all this other stuff to say.
Sir, I have another question.
It was an invitation, I guess.
Does E2F1 is the only target of this long non-coding RNA?
No. Actually, so what's interesting, there's actually, it depends on which assays you use, but it turns out that, so these are all known targets that were identified in the yeast 3 hybrid system. There were other targets found by RADA, and then there's other found by computation. So the question is, how do you know which one to study? In this case, E2F1 came out by all these different metrics, but an interesting property about long non-coding RNAs is that there's a large number of proteins that it interacts with, and that's actually one of the things that makes them hard to study, but makes them absolutely powerful and fascinating, you know, both as a subject of study, but also as a therapeutic target.
Thank you.
Let's give Dr. Novina another round of applause. And we have a little bit. Now let's move on to the third speaker of this session, Dr. Jonathan Abraham. Dr. Abraham is an associate professor in the Department of Microbiology at Harvard Medical School and a Howard Hughes Medical Institute investigator. He obtained a bachelor degree in biophysics from Harvard University. A biochemical science from Harvard College, a PhD in biophysics from Harvard University, and an MD also from Harvard Medical School. Dr. Abraham then completed a residency in internal medicine at Brigham and Women's Hospital and a fellowship in infectious disease in Brigham Women's Hospital, Mass General Hospital combined program. He launched his research laboratory in the Department of Microbiology at Harvard Medical School in 2017. The Abraham Laboratory used structural biology and molecular virology to study how viruses infect cells and replicate their genome. Most of the lab studies studies emerging viruses. Work performed in the lab aimed to bolster outbreak preparedness through basic structural and functional studies of the proteins that play a critical role in the life cycle of these viruses, including their entry proteins and nucleic acid polymerase, with a long-term goal of informing strategies for developing antivirals. Please join me in welcoming Dr. Abraham.
Okay, thank you very much for the wonderful introduction. I'll jump right in. Let's see if I can figure out how to advance slides. Okay, All right, so here I'm showing you a schematic diagram of a viral particle. The membrane of the particle is shown in gray. It's decorated by spike proteins. So now these are the business end that carry out the critical step of allowing the virus to attach to cell surface receptors. And so this is usually followed by uptake of the particle into, for the viruses we study, acidified endosomes, where low pH triggers a spike to undergo massive conformational changes that culminate in fusion of the viral and host cell membranes. So in the lab, we've set out platforms to discover receptors for viruses and also isolate neutralizing antibodies, usually from convalescent donors, with a bigger picture goal of trying to better understand how spike proteins can adapt to bind cellular receptors in different hosts, while also avoiding immune recognition by antibodies. So the story I'll be telling you about today is on our work on alpha viruses. These are positive sense RNA viruses that are enveloped. They're broadly subdivided into two groups, Old World and New World. This is based on their geographical distribution as well as their serological properties. An Old World virus that is famous is chikungunya virus. It causes rash and arthritis. A New World virus you might have heard about is Eastern equine encephalitis virus, or EEE, that causes encephalitis in humans and horses. And now, particular for the alpha viruses that cause encephalitis, there's no treatments or approved vaccines. Outbreaks can be quite devastating because the case fatality rate of infection can range from 30 to 70%. And many of the individuals who survive are left with permanent neurological sequelae, including cognitive challenges. Many of them end up requiring permanent institutionalized care once they survive the infection. And so why we're fascinating about these viruses is because of their broad host tropism. So here I'm showing you the example of the transmission cycle in nature. It really involves amplification in a reservoir that is usually birds. So mosquitoes will amplify the virus, spread it to birds that serve as amplifying hosts. And so this is called the enzootic cycle. And then infrequently, but significantly enough, there are these spillover events involving mosquito bridge vectors that will bite humans or horses, transmit virus, and humans and horses here are known as dead-end hosts. The dead-end bit comes from the fact that they will become viremic and can get sick, but the levels of virus in their blood is not high enough or sustained enough for long periods of time that mosquitoes biting those mammals would not be able to use this as a fresh source of virus. So the infection ends here. And so the spike protein for these viruses is synthesized as a polypeptide precursor that has two major subunits, E2 and E1. E2 is mostly important for binding to receptors, and E1 has a fusion peptide, and that's the bit that does the aspect of fusing the viral and host cell membranes, as I mentioned. So here, if you look at the surface of the virus, the membrane here now is shown in black, the capsid will be on the interior, and the spike proteins decorate this virus surface. And so this image doesn't really do a good job at describing the actual assembly of these viruses. They have icosahedral symmetry with T equals 4 triangulation number. And there's a total of 80 spikes on the virus that are formed from trimers of E2, E1 heterodimers. And so this will be an important point as I sort of move on through the talk. The other bit of background I wanted to cover is the important discovery of MXRA8, a cell adhesion molecule, as a cellular receptor for chikungunya virus. I would describe this as the first bona fide receptor for these viruses, described in 2018 from the lab of Mike Diamond at WashU. And this was based on the CRISPR-Cas9 screen and really opened up exciting areas in the field. And as with structures of alpha viruses, once the receptor is found, tend to sort of come out very quickly. There were multiple structures described, including X-ray and crystal structures. Here I'm just showing you E1 in gray, E2 in blue. The viral membrane would be at the bottom here. This is a side view, and this is a top view sort of looking down upon the virus. And the point I wanted to convey here is the receptor shown in pink is really hugging a cleft formed by the adjacent protomers of the spike protein, but not just binding to E2. And so what this means is that if one is looking to find new receptors, a system that would be appropriate for doing so would really have to recapitulate the proper organizations of the spike protein and ideally also the icosahedral symmetry so that all the trimers are properly assembled. And so many of these viruses are BSL-3 pathogens, however, so to work with them safely at BSL-2, we developed a single cycle system that involves taking the genome of a BSL-2 alpha virus, in this case Ross River virus, and then splitting it up into two fragments. So the first fragment here has the non-structural proteins followed by a capsid and the reporter. Oops, this is advancing. Good. And the other component has the spike protein that is provided in trans. So if you transfect cells with both the plasmid that has non-structural components as well as the second plasmid that have the spike protein, you get infectious virus formation for one cycle, but the virus cannot replicate. And so the reporter bit here is where we can put GFP or other proteins of interest if we're trying to monitor infection once the cells are infected. And so we developed a CRISPR-Cas9 screen that we modified to only target membrane proteins because we're interested in receptors. And so here we transduce guides targeting 4,400 membrane proteins in the genome at high guide redundancy, expose them to puromycin to have only cells that have guides. And then the key here is we're coming in with a virus that rather than expressing GFP as a reporter expresses CD20, which is a protein that's normally found on B cells, but is not found on the screening cell type. I'm showing CD20 here as these yellow hatch marks so that using agents developed, for example, to isolate B cells like max beads, we're able to deplete CD20 positive cells. And then doing this for a few cycles with viruses, we're able to then isolate cells that have guides that presumably render the cells resistant, so the cells are CD20 negative. And by next-gen sequencing, we can quantify which genes are really enriched as genes that were targeted by our guides. And so we did this first with Semliki-Forest virus, old world alpha virus that causes encephalitis in mammals. And we're surprised to find VLDLR, or the very low density lipoprotein receptor, as a top hit in the screen. Followed this was HSP9DB1, which is a chaperone for a protein called PCSK9, which is a master regulator of expression of LDLR-related proteins. And SCT3A also popped up, which we believe is important for VLDLR expression. And so showing you sort of a schematic view of what this looks like structurally, it has a long N-terminal ligand binding domain that comprises these LDLR class A repeats. These are cysteine-rich modules that are repeats of each other, but imperfect repeats, so there's a lot of sequence diversity. And it's really thought that the sequence diversity is what affords this family of proteins the ability to recognize diverse ligands, including lipoproteins, but also other proteins. And by screening other structural homologs of ApoE2 in overexpression studies, we found that both VLDLR and ApoE2, another LDLR member, were receptors for not only Semliki-Forest virus, but also for Eastern equine encephalitis virus. And so now we wanted to better understand this question that really first got us fascinated about these viruses, is whether or not the ability to bind LDLRs could explain the broad tropism of these viruses. I should mention that mosquitoes, even though they're separated by hundreds of millions of evolutionary history years from humans, also encode, because lipids are so important, LDLR-related proteins. There's one called lipofurmin receptor 1, or LPR1, which is expressed in the mosquito gut, for example. That is a close homologue of VLDLR. And so birds, of course, as well, will have these receptors, as well as horses. So we basically took cells that are resistant to infection naturally and then transduced in now forms of these receptors, including human, horse, bird, and mosquito orthologs for VLDLR, and then human, horse, and bird for ApoE2. And in all of our assays, whenever we tested these proteins, we saw they could robustly support entry by Semliki-Forest virus. The student on this project, Lars Clark, also wanted to find a negative control here. So he looked for C. elegans, the worm ortholog of VLDLR, which is also expressed in the worm gut. And even that one was able to support entry by Semliki-Forest virus. When we looked at EEE, we saw a slightly different pattern now. So EEE was able to recognize some orthologs, but not all orthologs. So this was really, to us, the first time that these viruses might differ in their way they recognize VLDLR. And so we're structural biologists. We wanted to look at the structure of the interaction. So we ended up trying to figure out what would be the minimal binding fragment in this 8 repeat containing protein here. So we put each of these as a single repeat onto the rest of the scaffold of the protein, which are these beta propeller domains, as well as these EGF-like domains. And our hypothesis going into this experiment was that perhaps the most membrane distal LA repeat here would be the one that would be bound by the virus. But when we tested this here, I'm showing you overexpression studies of various fragments from LA repeats 1 all the way down to 8, VLDLR being the control. We can see here in this case that it's actually multiple LA repeats that could work for Semliki forest virus. And if you look at Eastern equine, there's some differences again, where Eastern equine is able to bind to the first LA repeat, but Semliki forest virus does not. And then jumping to cryo-EM, I'm showing you here top views of the Semliki forest virus spike and the triple E spike. I'm showing you here now initial subunit. I didn't show you in the other ones, E3, which is a chaperone that we saw in our preparations. And so when we looked at structure of Semliki forest virus bound to a VLDLR LA repeat, we saw the LA repeat was actually binding to the E1 subunit here with domain 3 as a region. But when we looked at Eastern equine, that site had actually shifted by about 40 angstroms and was more similar to the MXR8 binding site, which is in clefts formed by adjacent protomers. So even though both viruses bind the same receptor, they actually do so by using different patches of their glycoproteins. And so why is this at the molecular level? Having available cryo-EM structures, we could look specifically at the determinants of binding. And the key here is that on the spike protein, all you really need are 2 basic residues that tend to form contacts with the acidic residues in the calcium binding region of the LA repeats. And similarly now, if you look at Eastern equine, you have a site on the E2 glycoprotein, the 40 angstroms away site, whereby you have 2 basic residues that are engaging the calcium coordinating acidic residues of the LA repeat. And the key now is if you look at sequence alignments, you'll see that Semliki forest virus E1 is really conserved to have those positions be lysines here, but that's not the case for Eastern equine. And if you look at Eastern equine, these residues are conserved across strains, but they're not sort of conserved at the site where SFV binds to LA repeats. So really there's a shift in where the virus is able to bind to LA repeats. And this mode of binding, I should just add, is very similar to how LDLR-related proteins interact also with their physiological ligands like receptor-associated protein here. So all you need is a minimum requirement of 2 basic residues that can interact with this calcium coordinating acidic residues on the receptor. And so for the first part, I've showed you that VLDLR and ApoE2 are receptors for Semliki forest virus and Eastern equine, that divergent receptor orthologs support alpha virus entry, and that viral spike proteins can have different binding sites for a same receptor. And now I'll sort of shift gears to tell you about the epidemiology of some of these viruses. So Eastern equine encephalitis virus has been around for some time, causes outbreaks every year that range from no cases to about a dozen cases. The largest outbreak in the last 50 years actually happened in 2019. I think there were 32 cases in 7 states. And this virus has garnered a lot of attention, again, because of the high lethality for the human cases. Now, another virus that many of you might not have heard of is called Western equine encephalitis virus. So this virus now is actually historically one of the most important alpha viruses in North America. It used to cause outbreaks involving hundreds of human cases and thousands of horses early in the 20th century. But for some unclear reason, it's done what our collaborators have called submergence. So rather than emerging, it's sort of gone away as a human pathogen, with the last case in North America really being in 1999. And the molecular basis for this was unclear. And I showed you that Eastern equine encephalitis virus binds to VLDLR and ApoE2. If you look at Venezuelan equine encephalitis virus, this is a closely related cousin to Eastern and Western equine. Mike Diamond's lab at WashU had shown that that virus binds to a protein called LDLR83, which is effectively almost like a truncation construct of our receptors here, whereby you have 3 LA repeats, and the first LA repeat is a site of virus attachment. But the third member of this family of viruses, Western equine encephalitis virus, did not have a receptor identified yet. And so we hypothesized that this would be an LDLR-related protein, but when we tested these 3 proteins in overexpression studies, they did not work for Western equine. And so we did a CRISPR screen, and what we landed on was, interestingly enough, a protocadherin. This is protocadherin 10 as the only sort of top hit that was significant. It's a protein that is involved in synapse development. It's enriched in the brain, but also expressed throughout the body, and it's also encoded by an autism-associated gene. So it made sense for this to serve as a receptor for an encephalitic virus. And when you look at how it's organized, rather than having LA repeats, it has cadherin repeats here. And these tend to form homodimers and trans as a way of orchestrating synapse communication in neurons. And so now we also had access to, interestingly enough, strains for the past century. So I mentioned that there are viruses that used to cause epidemics in the early 1900s. These are subdivided as lineage A here. And then each of these lineages have evolved over time to replace the prior lineages, if you think about SARS-CoV-2 variants, for example. And so that what's been circulating the most recently in 2005 are B3 lineages. And sort of an unanswered question in the field is observation that if you look at the virulence of the ancestral strains, they're actually quite lethal, killing 100% of infected mice. But if you look at the 2005 strain, for example, actually it's virtually non-pathogenic in animal models. Another tidbit I should mention is when we did our CRISPR screen, we actually used a 1971 strain isolated from a horse in Montana, but we had not tested additional strains. So the student on the project, Wan Yu, decided to test these strains and discovered, interestingly enough, that the ancestral strains all had the ability to bind protocadherin 10 from human and horses, but the latest 2005 strain actually seemingly had lost the ability to bind these receptors. And interestingly enough, that has us scratching our heads asking, so what receptors are these viruses then binding if they're not binding to protocadherin 10? Until we tested the bird ortholog, which is relevant to the enzootic cycle, and found that the bird ortholog could still serve as a receptor for the B3 virus. And that's sort of an interesting tidbit. I should add that birds will migrate away during the winter, of course, and so there's a hypothesis that there's an overwintering host for these viruses that can actually sustain prolonged viremia over the winter. And so when we tested the snake orthologs, snakes are known to be infected by these viruses, we saw that snakes, another relevant host for the enzootic cycle, that receptor actually still worked. And one question I get that'll preempt is people's like, how do mosquitoes bite snakes? Wan Yu taught me that apparently they bite the eyes. So usually I skip that detail because I think it's disgusting, but I figured I would share it now. But effectively, snakes are thought to be an important host as well. And the other surprise when we looked at the ancestral strains that are really hot in the lab, again, highly lethal in mouse models, they also had the ability to bind the same receptors as Eastern equine encephalitis virus. And so we're seeing quite a bit of shifts in the ability of these viruses to bind to receptors, which of course is very exciting for structural biologists to then go after. So Xiao Yifan did the cryo-EM structure of protocadherin 10 bound to the Western equine encephalitis virus spike. And quickly, by looking at sequence alignments, we can see here that there's a leucine in the E2 protein that in 2005 strains became a glutamine. And if you look at the structures, the receptor here is shown in orange, the spike is shown in blue. This leucine on the spike protein sits in the nice hydrophobic pocket here, and replacing that with a glutamine disrupts these non-polar contacts here, and would result in loss of affinity for the avian receptor, for the human receptor by the strains that have this polymorphism here. And so effectively what Wan Yu and Xiaoyi did as well is they showed that even though there's a loss of a contact here made by this 2005 strain with the avian receptor, that the avian receptor can compensate for this by making contacts elsewhere with the spike protein. So really the avian receptor is sort of a high affinity receptor for these viruses. And then we also looked at Western equine encephalitis bound to VLDLR, showing that the site is similar to what Eastern equine encephalitis virus binding site is. Here there's actually 2 LA repeats in the cleft, and again, different from Semliki forest virus in its binding mode. And so now we have these spike proteins, we have the structures, we have sequence alignments. So Xiaoyi and Wan, you were able to make predictions and look at a Imperial 181 pseudotype here that does not bind to human or horse orthologs. This is a 2005 strain, and show that a single mutation allows it to bind to the human and horse receptors. This is the Q149L mutation. And 2 mutations actually allow the virus then to bind to VLDLR and APOE2 and PCDH10. So even though we have these 2005 strains that are circulating and are non-pathogenic, presumably in North America, there are only a few mutations away from being able to bind the receptor. the human and the horse receptor. And so we also had information now of binding modes, and you can hopefully see from these slides that the binding site for VLDLR and protocodyrin-10 is the same. So if you take these receptor decoy proteins, is what we call them here, that have an FC domain instead of being membrane bound, and EC1 repeat here from the protocodyrin-10 receptor, that in vitro actually blocks access to VLDLR and protocodyrin-10. And when we looked at this with Scott Weaver's team at UTMB, our close collaborator, we saw that administration 6 hours before viral challenge with a protocodyrin-10 FC fusion protein was highly protective against, is that a mosquito? I'm teasing. Was highly protective against group A viruses here. And similarly, if you take the LA1, LA2 FC fusion protein, that's also protective against the ancestral strain. And so summary for the last part is I've told you that weave can bind to PCDH10 as a receptor on mammalian cells, that ancestral strains also bind VLDLR and ApoE-R2, and then structures clarify how E2 polymorphisms influence receptor dependencies. I skipped a part where we sort of describe also that, interestingly enough, as we were doing this work, in South America, Argentina, Uruguay, and Brazil, there was an outbreak of Western equine encephalitis virus in 2023, 2024. First time in 30 years, 200 human cases, thousands of cases in horses. But really, we could see very quickly once the sequence became available that they had L149, and we knew they would bind PCDH10, and we could also know they didn't bind VLDLR. So I think this information is hopefully useful for proactively predicting what viruses might do once they reemerge. And so the work was really done by Xiao Yifan, a postdoctoral fellow in the lab, Tan Yang, another postdoc in the lab, and Wan Yu Li. We have several close collaborations, including with the lab of Isaac Chu. I know Isaac is speaking later today, as well as Scott Weaver's lab at UTMB for the BSL-3 work. And I'll be happy to take any questions.
Thank you, Dr. Abraham. Any questions? If you have any questions, please raise your hand to the microphone.
Hey, Jonathan, great talk. I was always wondering about the encephalitis part. So you said the dead end host, like is the brain infection somehow advantageous for the virus in terms of spreading, or is it just by chance that those receptors are there?
I think it's mostly by chance. And actually, there's viruses that will infect humans and only cause rash and arthritis, like chikungunya virus, but it's binding into MXR8, which is a protein that's enriched in fibroblasts and joint tissues, et cetera. So I think for viruses that can bind to VLDLR, others have also shown since that maybe that's what's allowing transcytosis in the brain. You get seeding and then replication there, but it's really a dead end process because the host gets killed by that point.
Because avian doesn't bind to the brain ones.
That's a great question. Yes, for whatever reason, in avian ones, I mean, there are studies where they've tried to see what the impact is if there is an effect on viruses, but I'm not sure. Yeah, I mean, for the case of avian isolates most recently found, they will bind to avian PCDH10, but not avian VLDLR. But I don't know the answer to that one. It's a great question.
Great work.
Do the receptors cycle constitutively, or is this induced by the virus binding? And then how does the fusion happen? Do you dissociate the receptor from the viral glycoproteins before you actually get fusion?
Yeah, those are great questions. So I would say for the ones that are LDLR-related proteins, because those pathways are very well established and studied, this is presumably calithrin-mediated endocytosis. There's actually, for certain forms like VLDL and the LDLR, conformational changes that allows ligand release, where the LA repeats the same basic residues interact with the beta propeller domain, and that causes the receptor to close and release the ligand. So there we have a beautiful model for what might be happening with the virus. Fusion would happen pH-induced with the receptor falling off. Protocodyrin-10 is more interesting. It forms dimers in trans. What we can see is that the virus presumably can only bind the monomer of PCDH10 because, interestingly enough, the dimerational interface makes similar contacts to what the virus is doing. So there's sort of some molecular mimicry there. In our assays where we do life-cell confocal microscopy, it seemed that protocodyrin-10 mediated internalization was much slower than VLDLR-based ones, but we don't know enough yet about the cycling and the pathways. That's a great question. But presumably the dimer falls apart, the monomer binds, that gets trafficked, internalized.
Oh, thanks for the talk. It's very cool to see this kind of plasticity in terms of host protein and viral protein interaction. So do you have an opinion on like why this occur? Like, is it because the structural make it more poised to be this plastic, or is actually the receptor that it binds to just can easily bind to many places?
So
I wonder, are there other cases like other viruses that also have this huge variability on like how at which points of the capsids can bind to the receptors?
Yes, yes.
Yeah.
So I think the solution is most likely a combination of genetic drift, but also antibody-mediated pressure. The antibody pressure presumably here will be coming in birds, although it's hard because some of the sites are not well exposed. And so our hypothesis is the same as, I didn't show the mutations, but many of these mutations are like, E181 to lysine, which changes the charge as well as changing the residue that you would see in SARS-CoV-2 variants, all right? And so you're recruiting LA repeats because the minimum requirement requires only 2 lysines. And the bit about other viruses, there's explosion of data, obviously, on coronaviruses these days, where you'll have, coronaviruses that are like SARS-CoV versus MERS-CoV that will bind to ACE2 versus, DPP4, et cetera. And the way the receptor is binding to the virus sort of dances around. And presumably, I assume it's driven by mostly antibody pressure.
Okay. Yeah. I really enjoyed your talk. I was interested in the protocoherence. So they'll form homotypic or heterotypic binding across the synapse. So I'm wondering, does that increase the number of potential viral binding sites, and could that be a way for it to target synapses, for example? Is the infection enriched to synapses because of increased
We
potential for
have
binding?
not looked at that. The closest we did is, looking at mouse primary cortical neurons with Isaac's help, but we haven't done any imaging to see. The only thing I suspect based on the structure is that the dimer would have to fall apart, though, because the dimer itself would not be able to engage because it's occluding that surface. So presumably there's some free monomers diffusing, and that's what the virus is latching onto. Okay, thanks, everyone.
Okay, let's give him another round of applause. And
So we're now taking a 15-minute break. Please come back by 10:50.